In a series of lecture on bone tissue. And today we're going to go through and describe bone structure. We'll look at compact bone, we'll look at spongy bone, and then we'll begin to talk about how bone is made, of which there are two processes. So we'll look at that. Let's go ahead, excuse me, and begin with looking at the structure of compact bone, and then we will move into spongy bone. So first, compact bone. And you've seen this image many times now, but let's go ahead and look at it once again. Talk about some of the structures we're going to be looking at and describing. So the basic structural unit of compact bone is the osteon, which you can see pictured right here. In the center of the osteon is a structure called the central canal, and within that central canal we have blood vessels and lymphatic vessels. And I think I've mentioned this before, but typically where you have blood vessels, you'll also have lymphatic vessels in the body. And you'll learn about lymphatic system and the purpose of the lymphatic system in terms of its connection to the circulatory system in 40C when you do the immune system. So you have the osteon, and then surrounding that central canal are a series of concentric rings of matrix of bone tissue, and they form a ring around that central canal, much like a bullseye, where you have the center of the bullseye and then the circles around it. The structure internally, or the structure of the osteon in terms of the cell arrangement, is the osteocyte, which is contained within little spaces called lacunae. And then we get these small canals, canaliculi, that extend out and connect one cell with another cell. And if you recall, when we were looking at the cell types, we noticed that the osteocyte had these strange projections, and those are the projections that I talked about being inside that canaliculi. So those structures you can see right here, and those are the canaliculi. And so one cell is communicating with its neighbor through those openings and the extensions of the cell membrane into those openings. So basically what's happening is this is your source of nutrients. It's also the source that you can use to get rid of waste materials. So the cells closest to this will pick up oxygen, nutrients, and what have you. And then, like a bucket brigade, they're going to pass them from one cell to the next, and then the cells will be passing back their waste materials. And so there's this communication through those structures, canaliculi, from one cell to the next. And that means that the cells on the outer ring are not going to be starved for oxygen. They're not going to be starved for nutrients. Because remember, this is solid mineralized material, and there's no way that a cell living out here at the periphery is going to be able to survive through simple diffusion and getting its materials and getting rid of waste. So that's the basic structure of compact bone. Let's go ahead and put that into our notes. So we'll start with compact bone. So this is the structure of bone. Structure of bone. And we'll begin with compact bone. Compact bone, a couple of things that we can say about it. It contains few spaces, and as you saw in that image we were just looking at, there were not many spaces within that structure. It forms the outside region of bones, or more probably the outer edge of bone, the outer layer of bones, and the bulk of the diaphysis of long bones. Okay. Let's take a look at close-up look at that. So I don't have a bone that opens up on the inside, so I cannot show you the shaft portion, but I can show you this. As soon as we come into focus, there we go. We can see a nice spongy bone here. And if you look very carefully, the outer edge is a very thin plate of compact bone. As we move down into this region, which is the shaft, the diaphysis, then all of the edge basically would be composed of compact bone. Yep. So compact bone is designed to do a number of things. Protect, so it provides a protective barrier where we need it. Support, particularly the shaft, the diaphysis, being composed of compact bone as a supportive structure. And finally, to resist stress. As we already know, the basic unit of construction for compact bone is in fact the osteon. So let's refer to, or let's, so basic structural unit is the osteon. Okay. And it is composed of a number of structures. We'll start naming them, and then we'll draw a quick picture. So, number one, so-called perforating canals. Okay. And then number two, we have the central canal. Number three, we have concentric lamellae. There's a T. Let's do that again. Concentric lamella. And number four on our list is going to be the lacuna, or lacunae. Oh gosh, that's an N. Okay. And then we also have the canaliculi. And these structures right here, those four things make up the osteon. But we also have a sixth structure, which are the interstitial lamellae. Okay. And these are basically fragments of old osteons. And remember, it's important to keep in mind that bone is constantly being remodeled, so it's not surprising that as you remodel an area, you will leave behind some of the structures that were originally there and replace them with newer structures, or replace the removed things with newer structures. Let's take a look and see what we're talking about here. So if you look carefully at this drawing, so I have an osteon right here, I have an osteon right here, and then in between, filling up the space in between, is the interstitial lamellae. And these once were an osteon. So at one point there was an osteon here. When this got remodeled, that was left behind, and the new osteon was put in place. So not everything gets removed when we remodel. The material that's left behind, generally between the new osteons, is referred to as the interstitial lamellae. All right, so we've covered all of those structures right there. Okay, let's go ahead and draw a very quick picture to put into our notes and just identify these structures. Okay. So really quite simple. You identify the central canal and then a series of concentric lamellae around it, such as this. I generally put it in a rough pattern, and then I come in and fill in the details. Makes it easier. So that's that. And then we have these spaces here. And probably not going to draw everything, but enough to label it and identify the parts. Okay. And then we can draw in, and of course I don't have it here. What happened to this blue? That's it. So we're going to draw now little canaliculi. All right, let's go ahead and just label things. Add a little bit of color maybe to highlight some stuff. And there we go. And let's go. All right, so we're not seeing the perforating canals. I'll show those in a second, but we do know that that right there is the central canal. We also know that these structures here are the lacuna. We also know that these structures right here are the canaliculi. Okay. And let's see, what else have we got here? The concentric lamellae. So why don't we do this? Bring in a nice strong color here. And from here to here, we're going to identify that. And that's my concentric lamellae. Okay, and that's your basic osteon. Pretty straightforward. Easy, easy, easy. Okay, let's revisit that image. I want just to point out the perforating canals, because that's not shown in this image right here. So here we are, coming back to this. And these are the lateral connections of blood vessels. So this is the long axis of the bone here. So this is my long axis. And then we have these transverse canals. These are the perforating canals that connect one central canal with another central canal, as you can see right here in this drawing. Okay. All right, so that's the basic structure of compact bone. Let's talk about spongy bone. So spongy bone is very, very different from compact bone. And as you can see here in this drawing, this is an example of spongy bone. As you can see, there are no osteons. So right from the start, it's very different. No osteons. But we do have these complex lattice work of structures that are referred to as trabeculae. And within a cross-section of each trabeculae, we have the lacuna, we have the osteocytes, we have the canaliculi, But that's it. There is no other similar structure, or there is no structure similar to an osteon other than those things. This picture is interesting because we can see that there's an osteoclast, so it's breaking down the bone in this area. We can also see that endosteum, that layer of cells that surround the trabeculae, and these are going to give rise to the osteoblasts that will come in and remodel this area as we break it down, or as need be. So that's the basic structure of spongy bone. As I've shown you before, and I'll come back and show you it one more time so we can see it. There's a nice little portion of that in there close. There it is. There's that lattice work that we're looking at. Okay, so very, very complex. And remember, each of the surfaces that you're seeing here, all that lattice work is covered by a single layer of cells. That's the endosteum. So just as we saw in that last image. Of course, not now, because this is dead bone material, so that's all been removed and gone. But you can see that structure. Okay. All right, so spongy bone. Let's go through in our notes and identify the salient features. So, number one, no osteons. Okay. Instead, what we have is trabeculae. And this is a complex network of bone tissue with spaces between the trabeculae. Okay. The spaces between trabeculae is filled with red bone marrow. So all that space that we saw in those last few images filled with red bone marrow. The trabeculae contain, number one, osteocytes, within the lacunae, and the canaliculi. Nourishment is going to come directly from the surrounding red bone marrow. So unlike compact bone, there are no blood vessels within bone tissue. And just to emphasize that point, come back and look at this image. And perhaps we won't see it in English. There you go. No blood vessels. So all of this space that you see around the trabeculae is filled with red bone marrow. And so the transfer of nutrients and waste is happening directly into this space here. So that space is oxygen rich, has nutrients, it has the capacity to take away waste materials. There are blood vessels within that mixture of red bone marrow. So these cells are close enough to that outer surface in the trabeculae for that process of exchange to take place quite efficiently. Okay. So where is spongy bone found? Spongy bone is found in most, number one, so most bone tissue of flat bones, irregular bones, and in the epiphysis of long bones. So it's also the type of bones, so some type of bones. And I should have probably said examples is better. Would be hip bones, ribs, breast, sternum, vertebrae, and the end of long bones, of course. All right. So that's all we really need to know in terms of the structure of bone. Let's go through and identify sources of blood for bone tissue and the nerve network that is supplying the bone tissue. Okay, so we'll do that. And to do that, we've got a nice little drawing here. Take a look at this. So, does anybody recognize that bone? To me, it looks like the tibia, huh? Flat top with the two condyles at the top, so that would be forming the knee joint. All right, so this is a long bone, as you can see. Here's my epiphyseal line. And so we have the top of the portion, the middle section, and the long section, the last, the diaphysis, metaphysis, and epiphysis. All of that is being indicated here. And for each of those areas, we have blood vessels entering the bone. So in each of those regions, we have blood vessels and veins leaving at the same time. So the difference in the diaphysis as opposed to the other two regions is that we have one large opening through which the nutrient artery, well, they finally identified that as the nutrient foramen. Nutrient artery enters in here, it branches, and then it gives off sub-branches that enter into the bone tissue. And so you can see in terms of which arteries are supplying what part of the bone is very easy. So you can see here that the epiphyseal artery is going to supply the epiphysis, which is all of this region here, and extending a little bit down into the metaphysis, where the metaphyseal arteries are concentrating in this region here, the metaphysis, excuse me, and then the nutrient artery is supplying most of the shaft, or all of the shaft, I should say, also extending in through the bone and ending up blood within the periosteum, and also up into the metaphysis as well. So that's our coverage in terms of the blood supply. So blood supply. So for the metaphysis, let's start with the diaphysis. The diaphysis is going to be the nutrient artery. Also supplies portions of the metaphysis. Um, let's see. For the metaphysis, we have the metaphyseal artery. And I'm just talking about the arteries, but please note that for every artery there is also a vein named in the same way. And that's typical throughout the body. You'll find arteries and veins together. Maybe not the same exact name all the time, but they will be traveling together. And the metaphyseal artery is going to supply the metaphyseal region. Okay. And some of the distal region of the diaphysis. Okay. And the epiphysis we have the epiphyseal artery. And this is going to supply, obviously, the epiphyseal region and parts of the metaphysis. Okay. We also have, to finish off this story, we also have peristeal arteries. And they enter at many points along the perforating canals. And they supply the outer region of the diaphysis. Compact bone. All right. We've already talked a little bit about the nerves. So just a brief comment here. So nerves are found within the periosteum. And we have the sympathetic nerves and pain nerves. All right. So next, we're going to talk about the ossification process. Look at the ossification process and break that down. As I mentioned, there are two ways in which we can build bone, and we're going to, in this lecture, just deal with the first in that list. And then in the next lecture, we'll finish up with the second way that bones are constructed, and we'll talk about how bones grow in length and in diameter in the next lecture after this. All right, so let's talk about that. Let's talk about the ossification process. So ossification. Okay. Ossification is the actual construction of bone tissue. So it's the production of bone tissue. Part of ossification is mineralization, that is the laying down of the minerals, but the entire process that we'll be discussing, all of it is the ossification aspect of bone development. So production of bone tissue. So typically what you're going to find is that the embryo is composed of fibrous connective tissue, or hyaline cartilage, depending on what pattern of development you're going to have. And this is replaced with bone tissue. So ossification can actually take two different sort of routes, two different ways in which it can happen. You can start off with the fibrous connective tissue and then replace that with bone tissue, or you can start off with the cartilage and then replace the cartilage with bone tissue. And those are the two processes we're going to be talking about. So ossification generally begins around the 6th to 7th week of development. Okay. And there are two patterns. One is intramembranous ossification. And the second one is endochondral ossification. Those are the two patterns. We're going to look at intramembranous ossification first, and then in the next lecture we'll talk about endochondral ossification. As I said, we'll also talk about bone growth, both in length and diameter. Okay, so let's begin with the intramembranous ossification process. We don't need that. All right. So, what is happening here? Bone tissue is going to replace a loose connective tissue also sometimes referred to as fibrous connective tissue. So basically we're going to build a small version of the bone that we want to make out of this loose connective tissue or fibrous connective tissue. And then we're going to go in and replace that with actual bone tissue. Okay? Some examples of where you can see this are the fontanelles. Okay. Of that fibrous connective tissue. Those spaces then are going to be replaced with bone tissue as the child grows. Okay. So let's start and just describe this in a step-by-step way. And we'll start with number 1, and we'll just keep on going until the process is finally complete. Then we'll take a look at some images. The textbook doesn't have any really particularly good images of this process, but we'll look at what the book has to offer and talk about these steps. All right, so we're going to get down in our notes first, and this first step is going to be the development of the center of ossification. Okay. And this involves a couple of things. The first step is that mesenchymal cells will cluster together. And that ability to start clustering together within a centralized location is due to a signaling molecule. So chemical messengers signal that development. And a lot of what we're going to talk about is driven by a complex environment of molecules which are directing the cells to behave in certain ways. And so there are chemical messages that are being produced within an area that cause the mesenchymal cells to cluster together in that area, and then they will differentiate. Again, differentiation, a change in the nature of the cell, is going to be initiated by these types of molecules we've been talking about. So differentiation happens, and they become osteoprogenitor cells. Okay. Then they become osteoblast. Okay. Now remember, this is happening within that, or on that surface, that surface of fibrous connective tissue. So the very first step is actually listed up here. I don't think I made that clear enough. So the bone tissue is going to be replaced with loose connective tissue, and the fontanelles are an example of that. So you're laying down the pattern initially with this fibrous connective tissue. Then these steps that we are talking about are going to occur. Okay, so development of the center of ossification. And so as this happens, then the osteoblasts begin to produce bone tissue. Okay. And some osteoblasts become surrounded by bone matrix. And thus become osteocytes. Okay. And then this process is, as this thing that we've described, these series of steps occurring, what we're producing is the ossification center. Formed through these series of steps just described. Okay. Now, it's important to understand that if you have a bone, say the parietal bone or the occipital bone, let's use the occipital bone. These ossification centers, there's not a single ossification center. There will be many ossification centers. So let's just kind of try to put that into a picture form so we can understand it. So this is the posterior view of the occipital bone. Very small, of course. There's not an adult size. So this is basically what you're seeing here, and let's just do this in yellow, is the fibrous connective tissue. So that is our fibrous connective tissue. And what you're going to get is these ossification centers through the steps that we've just described happening. So you're going to have a region here. You'll have a region here. And I don't know if there is a pattern in which this occurs in the bone. I've never researched that thoroughly. But you're going to get a series of points that are going to be the ossification center. So these are all ossification centers. And what's going to happen is they will eventually grow together to create a single bone. Now notice that this pattern that we're talking about lends itself. So let's pretend for a moment that for some reason the region that we see right here, which I'll highlight here, let's say that's the extent of where that ossification center is going to grow until it meets these neighbors. But if this region that we've outlined in red never fully integrates with the rest of this, then you're going to have a sutural bone. This will end up being a sutural bone. If that border between these areas of growing bone tissue never completely melds together, you're going to have a separate bone here. That would be a sutural bone. So that's why we see the sutural bones in the skull, because the bones of the skull are developing with this pattern that we're describing here. Okay, so that's the ossification center. Whoops. Center, forgot that, is formed through these series of steps. The next step in the process, the number 2, is going to be calcification. That's a C. Calcification. And as you know, calcification is the deposition of the mineral salts. Okay. So what we're forming here is patterns of or areas of differentiation, cell differentiation. They're laying down the fibers that we discussed in the last lecture, the collagen fibers and all the other proteins that we've discussed. And then as that process continues, we're going to do this. We're going to get the calcification process happening. So what's going to happen is we're going to secrete, or secretion of, extracellular organic materials. Okay. Stops. Secretion of extracellular organic material stops. So we've laid down those fibers, and once they're there in place, they're going to stop, okay? And that is when the osteoblast becomes an osteocyte. So osteoblast then becomes the osteocyte. Okay. So let's see if I can draw this in something that makes sense. So this is going to be a very poor, and then we're going to have another one here. These are, and we're going to come in and fill in so that so that's an that is going to be an osteocyte. And at this point, they're trapped in the organic matrix, not necessarily the, I'm going to do this, not necessarily the calcified salts yet. So they're trapped in a complex web of fibers. And that is the point at which they can be called osteocytes. Okay. So does not necessarily mean that we actually have the calcified material surrounding them, but they are surrounded by this very complex web of proteins. So now we can describe these as osteocytes trapped within the protein fibers. And now what's going to happen is we'll start to lay down the calcified salts, and it'll just fill in the spaces. But they're already, you'll notice that the pattern of that cell, that those with the extensions and creating space for those extensions to touch one another, is already there before we added the mineralized salts. Okay. So you're going to see these extend the cytoplasm. Processes into what is now going to be called the canaliculi. Okay. And then within a few days, mineral salts are deposited onto that complex web of fibers. Okay. So coming back to our picture, what you will begin to see is the deposition of the mineral material, forming crystals on initially on the fibers, but as the crystals are forming, they will also form upon themselves, and we will get then a pattern in which these cells are completely covered by, or surrounded, I should say, by the bone material as we know it. Okay. This whole thing that I've just talked about is calcification. Oakley dokley's. The next step is then the formation of trabeculae. Okay. So the mineralized process, or mineralization, I should say, that's probably a better word. Mineralization process forms trabeculae. Okay. And this is going to create or produce a network of bone tissue, which we can now call spongy bone. The connective tissue that is associated with the blood vessels within this area differentiates into red bone marrow. So there's blood vessels within these areas that we're building this bone, and there is connective tissue associated with that, and that is going to change and become what we call red bone marrow. It is a complex problem, or it is a complex process, I should say, and there is a lot we're leaving out, which is really not necessary for our understanding, especially for this class. But as you can see, it is quite complex, and we haven't even talked about all the different signaling molecules that they've discovered, which is directing this process. Finally, the fourth step is development of the periosteum. And probably we should have written that both things are happening. So it's location, really, that is determining the name. So periosteum is on the outside. Endosteum is going to be on the inside. But nevertheless, both those structures have to actually be built and formed. And so what's going to happen is that as trabeculae are forming, mesenchymal condenses at the outer surfaces of the bone and becomes the periosteum / endosteum, again, depending on location. Okay. For the outer edge of the bone, for the outer surface of the bone, so the outer surface of the bone, compact bone is produced under the periosteum. And much of the newly formed bone will be remodeled. Over time as the bone matures. Okay, so, when I first heard this idea that, we're going to build something, we're going to spend a lot of energy, a lot of resources to build something, and then we're going to turn around and remodel it on a constant basis. Why in the world would we want to do something like that? The answer is that bone tissue, as good as it is, as strong as it is, as resilient as it is, over time will start to break down. It will start to age. And I'm not talking about you growing older. I'm just talking about something that is being used and used and used and used constantly tends to wear down, just like anything that you own. When you use it over time, it tends to break down, and you have to either replace it or fix it. And of course, you're in a living body here, so you're not going to replace your body with another body. So what has happened is there has been an ability of your body to constantly look at in this case, bone tissue, finding places where things need to be fixed, where things need to be changed, and that's a constant ongoing process within bone tissue. So again, much of the newly formed bone will be remodeled over time as bone matures, and that's where we get the interstitial lamellae. That's the leftover or the remnants of places that have been remodeled and new material has been added. So the type of bones that would experience this process, we already talked about this: the skull bones, parietal bones, temporal bones, the frontal bone, the occipital bone, all of those bones that are part of the skull. The scapula and the clavicle, believe it or not. So when you look at the clavicle, you would say, long bone. It should have the structure of a long bone. In fact, it does not. The clavicle has a very thin layer of compact bone around it, but if you open it up, there is no medullary cavity. It's just spongy bone and throughout its entire interior. So it is built along these lines that we've just described. Okay. So let's talk a little bit about bone remodeling, since we've discussed this. And how does that happen? So bone remodeling. Okay. And what we're going to do is we're going to create or produce something called a bone remodeling compartment. And I'm going to draw that for you. And this has been abbreviated to bone remodeling compartment. That is the... So where is this being produced? When the need for bone reabsorption is sensed by the osteocytes. So again, we're getting back to the osteocytes sitting in the bone and being able to measure changes in the bone because they're sitting within that material. Then they will send out signals. These are chemicals to lining cells. And here we're talking about the endosteum and or the periosteum. Okay. These living cells then, or these lining cells, I should say, will retract from the bone surface to form the compartment. So what happens is the lining cells will retract to form the bone remodeling compartment. That is, that they will retract from the surface of the bone. Okay, so let's take a couple of pictures first, and then we'll go into this. And I'm going to use this image here. It's already been kind of nicely labeled and kind of lays out exactly what we want to talk about here. So this is trabeculae. And let's say for whatever reason this needs to be remodeled. And the osteocytes within the trabeculae are experiencing some sort of stress or some sort of signal that tells them that something needs to change. They'll send out signals, and these lining cells will actually form a bubble. They will retract and form a compartment over the surface of the bone. And that's what we're going to look at. So that's what we're going to identify. So get out your artist hat, get your pencils, your easel, your paint brushes, whatever you need. And we're going to draw this and label one of these bone remodeling compartments. So let's do that. So first of all, we're going to just draw the surface of the bone. And I would suggest kind of doing it in lightly here, just so you kind of know where you're going. Okay. And then we're going to start bringing in the cells that are the lining cells. And let's go ahead and we'll do that like this. So these are the cells that are forming the lining. And then they're going to form this. And here's where you want to really kind of just rough it in so you know where you're going. There you go. Now you kind of... So in this case, we're talking about the endosteum, right? We're talking about the single layer of cells that are sitting on top of the trabeculae. They've gotten a signal, and they've lost contact with the surface of the bone and created this compartment in the bone. Okay. And that color wasn't the best color to choose, so I'm going to go ahead kind of highlight things so we can pops out a little bit better. And of course we have the nuclei. These are living cells. Okay, there it is. All right. Now let's go ahead and finish in or fill in on the bone. So this is bone, as we've said. And let me pick out a good color here. Something that pops. So let's do this. I think this might be work here. So this is my bone material. Okay. That's bone. And we'll just... my bone. So this is bone. And of course, what we know is that we have these cells, the osteocytes, that are in that bone. And I'm just going to rough those in pretty undramatically. Okay. These are the cells that have detected the need for change. And they are releasing these chemicals that are initiating all of this change that we've been talking about, the remodeling process. Those are my osteocytes. Okay. So, osteocytes. And one last structure we have to bring in, and this is going to be, I should have left a space there, but that's okay. We can use our imagination. I'll do this. Nice, fill that in with some nice color. We'll label it in a second. We're just having a good time drawing now, actually. Okay. So this is going to be the capillary. So this is a blood vessel. So a capillary. And we're going to have some cells in there, which we're going to draw in here. These are being delivered by the blood vessels, and these are the osteoclast precursor cells. The blood delivers them. Why? Why is it the blood that's delivering those? Because these cells, the osteoblasts, originate from red bone marrow, so they have to be delivered through the circulatory system to this site. Okay? All right. Now, once here they leave, so they enter into the compartment, and there are signaling chemicals. That are being delivered to initiate change. Let's do, I don't know. I need to resupply my. Do that like that. So signals are being released by the osteocyte. And these precursor cells then receive that signal and they become osteoclasts. And they settle down on the surface and they begin to work their magic on the cell surface, breaking down bone. So the bone surface is being eroded away. At the same time, these same cells are also delivering another message, and this message is being sent to these lining cells, and that message is going to cause the osteoblast precursor cell to become an osteoblast. So another signal. And this is going to cause the formation of osteoblasts. Okay. And so we get then a cell, we get cell division, we get the formation of the cell, and it becomes then cells that are going to be adding new bone. Okay. And this is the osteoblast precursor cell. And that's how it works. This is happening. This is microscopic. It's very small. And that's the process by which bone is broken down, reabsorbed, and then replaced with new bone material. These cells are orchestrating that whole process by producing these signaling molecules that bring in the osteoclast and produce the osteoblast from the lining of the, in this case, the ostium, and replace that material which is being taken up by the osteoclast. All right, ladies and gentlemen, that we're going to call it a day here. The next lecture I'm going to deliver will be on the last, or the second, endochondral ossification. We'll go through that step by step. It's a slightly more complex process. We'll talk about that. We'll develop a bone through that process. And then we're going to look at the growth of bone, growth in length and in width. And then we'll have a little bit to say about the hormones that regulate the process, and we'll be done with this chapter. So I will see you in the next lecture. Tally-ho.